All right, well, welcome to lecture number 5, which is going to be all about cells. So cells are the primary unit of life. So we talked in previous lectures about atoms, then we talked about organic building blocks, then we talked about biomolecules. So now we're going to talk about cells. So cells are basically these containers where all of biological life happens inside of. So basically an entire unit of life has to be within a cell. So we'll talk about the different characteristics of cells. We'll talk about prokaryotic cells like bacteria versus eukaryotic cells, which is the cells in our body. We'll talk about different features of cells, some processes that go on in cells, some of the structures that make up cells, and a lot of the different organelles that are involved. And so these are little compartments within the cell that do specific functions. So within that, we'll pepper in some things about diseases that arises from different cells and different processes that they're involved in. So again, it's a lot of information. It's all about cells. It's a big topic. You can teach an entire course just on cell biology, and you can take that course very easily. But overall, you just need to sit back and sort of learn about cells, which are the fundamental unit of life. So I hope you enjoy the lecture number 5, cells. So today we're going to be talking all about cells. So the importance of that is that cells are the primary unit of life. So cells are the most fundamental that basically a living thing can be and still be considered a living thing. So the cell is therefore the fundamental unit of all life on Earth. Every single living organism on the planet is composed of a cell. So that's cytoplasm surrounded by a lipid membrane. So this is a lipid membrane surrounding the internal environment, which is cytoplasm, that contains biomolecules that we've been talking about for the first 4 lectures in this course. So these biomolecules are contained within the cell, and that's how that permits basically the fundamental processes necessary to sustain life and to reproduce. So we'll be talking all about what a cell is, what the modern cell theory is, and what the different processes, the different structures within the cell, and the different processes that the cell does in order to sustain life. We'll be talking about that throughout this entire lecture. So in your book, they talk about modern cell theory, and there's basically 6 points about modern cell theory. So this is 6 points that all cells have in common, and that sort of outlines their importance to life. So number 1 is that all life is made of cells. So all known living things are made up of cells. So most organisms, most species on the planet consist only of a single cell. However, other organisms are formed from our multicellular organisms. So it's a complex interconnection between a bunch of different cells. So the cell distinguishes living things from non-living matter. So does it have these properties within the modern cell theory? Is it made out of a cell? If it is, then it's alive. If it's not, then it isn't alive. So we have some examples of a bunch of different cells here. So here's a single cell eukaryotic cell, Toxoplasma gondii. Here's a amoeba, which is also a single celled eukaryote. Here's a type of bacteria right here. Here is a neuron, which, as you can see, is a very different in morphology than other cells. Here is another single celled eukaryote, and here is another bacteria, Vibrio cholera. So all life, all living things are made up of these individual units called cells, even though they may differ quite a bit in their size and their shape. Now, point number 2 in the modern cell theory is that cells form fundamental functional units. So a cell that's basically the structural and functional unit of all living things. So it's the smallest unit that can actually sustain itself and generate all the biochemical processes that we've talked about previously and we'll talk about in the future to sustain that individual cell and to pass on its genetic information to other cells. So each of these individual cells right here, there's a lot of different kinds, are individual units. So they are fully functioning, not necessarily independent, but they can function independently and sort of take care of their own processes. So you cannot have half a cell that's alive and functional. That's what we mean by it's the fundamental unit of all living things. So the cell is as basic as you can get and still be alive. So point number 3 in the modern cell theory is that cells reproduce. So all cells come from pre-existing cells by division. So this may seem pretty fundamental or like pretty, not too difficult to understand, but it was not known for many, many years. And a lot of top scientists in the world believed that small organisms like this just sort of spontaneously generated from inorganic matter. We know now that that's not the case. All cells come from division of another cell. So we'll talk about the difference between prokaryotes and eukaryotes in a bit, but prokaryotes, which are bacteria and archaea, they will reproduce by something called binary fission. And so that's outlined right here. So these are more simple cells, and their DNA is just floating around in the cytoplasm for the most part. It's organized a little bit. But basically, they reproduce that DNA into 2 strands of DNAs, and then these 2 sets of DNA segregate, and then they split off their cell membranes, and that results in 2 separate cells. So that's how bacteria like this will replicate. So they'll do it very quickly. And this is going to basically end up being 2 cells, whereas it was just 1 cell only a few, like a half an hour before this. Eukaryotes are a bit more complicated. So they divide through 2 different ways. So most eukaryotic cells divide by a process called mitosis, which is shown right here. And then some your reproductive cells basically divide by meiosis, which is a modification of mitosis. So you have a diploid cell, which means it contains 2 copies of each chromosome. It also replicates its chromosomes. Then these line up in the center of the cell. The half of them go to one side, half of them go to the other side. That cell membrane splits, and then now you have 2 different cells. Meiosis is very similar, except that you ultimately result in 4 different cells, and each of them carry half of the genetic information. So that actually will make sperm cells and egg cells that will eventually join together to form a zygote. So mitosis and meiosis. So in each of these processes, and this is sort of outlined right here, you can see a bunch of different eukaryotic cells, all in very different stages of replication. But all of these cells, so in each of these processes, either binary fission, mitosis, or meiosis, DNA is replicated and passed on to what are called daughter cells. So those are the 2 cells that come from 1 cell through replication. So all cells reproduce and come from pre-existing cells by division. You can't just get a cell that pops up out of inorganic or even organic material. So modern theory point number 4 is that cells contain DNA, which is sort of related to the previous slide. So cells replicate, but importantly, these cells also contain hereditary information. So they contain a DNA record in terms of that DNA genome that is passed from one cell to another cell during cell division. So we saw when cells replicate, they always have to replicate their DNA before they can split off into 2 daughter cells. So the DNA in the cells, of course, as we discussed in previous lectures, contains the genetic information that is expressed in the form of specific proteins. So the DNA will be transcribed into RNA, and then RNA will give the genetic code, the code to build specific polypeptide chains for the form of proteins. So cells, when they divide, they will inherit this DNA information. So again, we have prokaryotic cells over here. So you can see this blue is the staining of the DNA in these 2 cells. And you can see that they both, of course, contain that genetic information. And then here are some eukaryotic cells like we saw before. So this genetic information is actually contained in the nucleus, And then when these divide, as you can see, this one's in the process of starting to divide right here. They will pass that genetic information on to any cells that arise from that cell. If there's no genetic information in a cell when it replicates, that cell is just going to die. It's not going to be able to have the instructions to produce the proteins to do all of the process that the cell needs to do in order to survive and for itself to reproduce. And point 5 of the modern cell theory is that all cells are basically the same in composition. Now, there's caveats to this, of course. Different cells are different, just like it sounds. You know, they have different sort of functions and roles and needs for those cells to survive and reproduce, but they're all composed of basically the same overall structure, and they're definitely composed of the same biomolecules for the most part. So a prokaryotic bacteria, shown right here, a eukaryotic single-celled organism like an amoeba right here, and then a complex neuron, your eukaryotic neuron right here. All of these different types of cells are made up of, for the most part, the same proteins, nucleic acids, carbohydrates, and lipids. So the concentrations and the types of these different molecules may vary, but all these cells have, for instance, a lipid membrane. They all have a DNA genome. They all make RNA. They all make proteins. They may make different proteins depending on whether you're a liver cell or an E. coli cell or a neuron, but they all make proteins, and actually they all make the most fundamental proteins. They all make the sort of the same way, and they may use different carbohydrates, but they all use carbohydrates, and most of them use the same ones. So they also have the same ions and the same small molecules that are involved in most of their processes. So we've said it before, but what's true of E. coli is true of elephants. I forget who said that, but it really is true. So each one of those E. coli cells is basically doing the same biochemical process as the neuron in an elephant brain. So cells are basically the same in their composition. And that brings us to the sixth point of the modern cell theory, which is all energy flow, basically metabolism and the biochemistry of life, that all occurs within cells. So things like metabolic processes, genetic information processing, cell signaling, ion transport, trafficking of different molecules, and the degradation of molecules, and all the other processes that make a cell function, they all occur within the confines of the cell. So they may, they may send molecules, bring molecules in and out of the cell. They may react to signals that are coming from outside the cell. But all of these processes are occurring within the cell. There's basically very little biochemistry that's happening outside of cells. In terms of energy flow, all cells are basically in a struggle against entropy. So cells are very organized, ordered molecules, and you have to use energy for that to happen. The whole universe, basically the laws of thermodynamics say that the universe tends toward disorder. So that is a law of thermodynamics that cells are breaking, and they're doing that by putting energy into the system and increasing disorder elsewhere in the terms of heat and everything. So things like the citric acid cycle, which is a metabolic cycle shown here, that occurs within cells. Genetic information processing like DNA replication, transcription, and translation, that all occurs within cells. So cell signaling. So even though these signals may come from outside of the cell, those signals, the function of them are to make these signaling pathways that will change the overall status or function of or metabolic state or something involved in that cell. Same with processing of ions. So they may move ions in and out or out of the cell. That is going to occur within the cell, within proteins in that cellular membrane. And also like bringing in molecules, bringing in raw materials. These are brought in from outside the cell and then processed within the cell. So all the energy flow in cells, so they have to be able to take energy in from the outside of the cell and then process that within the cell itself. So we just talked about how the cell is the fundamental unit of life. So where did all these cells come from? What was the first cell? Well, it's not exactly known how that happens. So that's an active field of research that's currently being investigated, and sort of there's a lot of hypotheses out there on how that could have occurred. But the earliest evidence of cellular life has been shown to be at least 3.8 billion years ago. So that was only 750 million years after the Earth was formed. So and the evidence from this comes from basically fossilized remains called stromatolites, which are fossils that arise from the fossilization of these cyanobacteria. So these are very simple forms of life. Well, not so simple, but these are the earliest forms of life that we have evidence at 3.8 billion years ago. So cells arose quite early in Earth's history. So how that happened is not exactly known. So we've talked a little bit about it in the previous when we were talking about organic building blocks, but basically the formation of a membrane and then the processes necessary for a cell existing within that membrane is basically all you need to form a cell. So it's been theorized that there's three things that you basically need to have a cell. So you need that membrane enclosing, they have to have a membrane enclosed environment capable of copying genetic molecules. So you have to be able to pass on that heritable information, so in the form of DNA, like we just talked about. So they have to have a way to copy this and pass that off to other cells. They also need to be able to catalyze chemical reactions. So we talked about that issue before. So you need proteins to catalyze chemical reactions and to copy genetic information. But without the genetic information, how are you going to make those proteins? And one of the ways that people sort of bridge that gap is through the RNA world hypothesis, which we talked about earlier. And then 3, they have to be able to transfer environmental energy to usable chemical forms. So cells have to have some way to use energy in their environment in the terms of, well, now it's oxidizing carbon-based molecules, but they have to be able to turn that into a chemical form that they can use for the processes within the cells. So cells are very, very old, and we have evidence of 3.8 billion years ago at least. But how they arose, how they physically arose on planet Earth is a subject of active debate and investigation. So the most fundamental part of a cell, I would say, is the cell membrane. So all different cells have a plasma or it's a cell membrane. It's also called a plasma membrane outside of the cell, and made out of lipids and proteins and other molecules, but primarily lipids. So the cell membrane actually is necessary to enclose the cell content. So it provides a barrier between the environment on the outside of the cell and the environment on the inside of this cell. So things like lipids and gases can move through that membrane, but importantly, very large or polar molecules do not pass freely across that membrane. So they have to be basically transported across, or basically diffusion will take over and eventually it'll sort of even out. But that lipid membrane provides a barrier around the cells. So you can see here the plasma membrane or the cell membrane forms a barrier around each of these individual cells. And so that encloses the internal contents of that cell away from other cells and away from the external environment. And so we already talked about lipids spontaneously form these vesicles when you put them into water, and you can get them if you just take a bunch of lipids that are polar, so they have a hydrophilic head groups and a hydrophobic tail groups. If you mix them in water, they can form these vesicles here, or these bilayer membranes, where there'll be an outside environment here and an inside environment here. So that's the most fundamental definition of every single cell that exists. So the most important lipids in the cell membrane are phospholipids. So we talked a bit about these in lecture 3. And so these lipids, there's a lot of different types of them, and some of them are shown here. So this is phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol. So, but what they all have in common, of course, is that they have a polar hydrophilic head group. See, all of them have charged groups with the phosphate and another charged group in their head region. And then they have hydrophobic tails, all of them. And so this will spontaneously form a lipid bilayer like you see here. So these positively charged groups are interacting with the outside environment. These positively charged groups are interacting with the inside intracellular environment. And then you form this lipid bilayer from these hydrophobic groups, which can interact with each other, these non-polar hydrocarbon tails. And so here's another diagram of what that would sort of look like in a space-filling diagram. So this is what's going to be impenetrable to charged and polar and water-loving molecules, basically. So gases, things like gases can slowly sort of move across the membrane, and also lipid molecules will eventually move across the membrane. But anything that's charged, there's going to be a barrier there where it's basically just going to bounce off that membrane. So those are the lipids. So phospholipids are highly important to form that cell membrane around each of your cells. Now, another important lipid component in cell membranes, specifically animal cells, is cholesterol. So cholesterol is a lipid steroid molecule, and it is considered a lipid because it is hydrophobic. So cholesterol has this long hydrophobic hydrocarbon group right here that's composed of 1, 2, 3, 4 fused rings that are made entirely of hydrocarbon molecules. And then up here, it has this polar hydroxyl group, this single polar hydroxyl group. And so cholesterol is really important for a lot of reasons. It's the precursor for all the other steroids and for vitamin D and also for bile salts, which we didn't really talk about. But it's also really important in animal cells because it actually can incorporate into these lipid bilayers in your cell membranes. So this is a diagram of that right here. So we have these cholesterol molecules right here. And so that lipid group is going to interact with these hydrophobic tails in the lipid bilayer right here. And then that hydroxyl group can actually interact with the hydrophilic head groups. So that's very convenient. And what this does is it's basically, when these are incorporated in, they push away these other hydrocarbon tails in this lipid, this phospholipid bilayer. And that will sort of increase the fluidity of the cell membrane. So some cell membranes are more fluid than others, and your body has to have ways to regulate how fluid cell membranes are. So cell membranes can't be too hard, less fluid, and they also can't be too fluid because they would just sort of fall apart. So cholesterol is a way that animals can sort of control the fluidity of their membranes for different purposes. And that's why too much cholesterol in your diet can lead to things like atherosclerosis, which is a hardening of your sort of membranes in some of your tissues that can cause heart disease and clots and things like that are not so good. But cholesterol is obviously integral to your body because it's so important in the plasma membranes of each of your cells. Now, what's interesting about lipid membranes or cell membranes or plasma membranes, whatever you want to call them, is that they are way more complex than we just sort of showed. So they are primarily made of these lipid bilayers, but really importantly, cell membranes are made of a combination of lipids and proteins. And so as you can see here, so that gives us what's called a fluid mosaic model of the cell membrane. So it's this lipid with these embedded and associated proteins as well. So there's a lot of different types of proteins that can be in the membrane. So you can have a lot of proteins that are integral in the membrane. So they have basically a part of the protein inside the cell, a part of the protein outside the cell, and then they have a region that's embedded within the membrane. So the outside of that would have hydrophobic amino acids sticking out from it, so it can interact with the hydrophobic tails in that cell membrane. You can also have peripheral proteins that are just sort of associated with the membrane. And you can also have proteins that actually have lipids sticking out of them, and then that allows them to associate, to sort of embed themselves or anchor themselves into the membrane. Now, these proteins and these lipids on this membrane can also be decorated with carbohydrates. So we talked about glycoproteins very quickly in a previous lecture. So you can have a lot of glycoproteins are embedded within the cell membrane. And also you can have glycolipids. So the lipids within the membrane can also be have glucose or carbohydrate groups on them. And then this model right here also shows these cholesterol molecules within the lipid membrane. So this is what's called the fluid mosaic model. So it's a way to visualize the cell membrane, which includes not only phospholipids, which are the main component shown here in blue, but also these proteins that are either embedded in the membrane or associated with it, these carbohydrate groups that are attached to the lipids and to the proteins, and also the cholesterol within the membrane. So you can see that it's a very diverse and highly complex sort of structure. And so the proteins are really important for the membrane, and we'll talk over and over about membrane proteins, but they provide functionality to the membrane. So if there were no proteins in the membrane, it really wouldn't do too much. It'd just be a barrier between the outside and the inside. So these proteins in these membrane allow it to do things like signal transduction, so to get signals from the outside of the cell to change the environment and the inside of the cell, and also to move molecules from the inside and the outside of the cell. So the types of proteins that are in the membrane and the percentage of proteins that are in the membrane compared to lipids vary between different cell types. So different cell types, based on their function in like multicellular organisms, will have different types of proteins on the outside of those cells. And what types of proteins are on the outside of the cell may actually define what that cell type is. So if it's a liver cell, it's going to have certain proteins in it in the membrane. If it's a neuron, it's going to have different proteins in the membrane. If it's a cardiac heart cell, it's going to have different proteins. And then the percentage can change as well. So most plasma membranes in most of the cells of your body, like your basic liver cells and stuff like that, those are about 50% protein. And things like myelin sheaths, which we talked about in the previous lectures, so those are those membranes that go around the neurons to help those nerve impulses go faster. So if there's a breakdown in that, you can get something like multiple sclerosis. Those have very low protein composition, so only about 25%. Whereas your mitochondria, which are sort of, we'll talk about those in a bit, but those also have lipid membranes around them, but they have a huge amount of proteins in them because they do so many, important metabolic activities. So in mitochondria, those membranes can have like 75% proteins in their membrane. So it's very important to understand that the lipids are the main component of these cell membranes, but that proteins and carbohydrates and cholesterol also play very important roles. So with that fluid mosaic model, we kind of just touched on the mosaic part. So the cell membrane is made out of lipids, proteins, carbohydrates, cholesterol, which is a type of lipid. But we didn't really talk about the fluid part. The fluid part comes from the fact that the lipids and most of the proteins in your cell membranes will diffuse laterally very quickly. So if you have a lipid in this position, it can move side to side through that membrane very rapidly. And so we know that from a lot of very clever experiences called fluorescent recovery after photobleaching, or fluorescent recovery after photobleaching, FRAP. You don't need to remember that, of course, but it's a cool experiment. So what you would do is you'd basically take a lipid in a cell membrane or a protein, and you'd label it with green fluorescent protein. So this is just, you would basically attach a protein on here that fluoresces green, so the color green. And you can see that under a microscope. So then you could take a cell, and then you can hit that green protein in a specific spot with a very strong light that'll basically bleach out all the colors. So there's no more fluorescence in that area. You can see how it's like really white at this position. So all you have to do then is just wait and see how quickly these other lipids or proteins flow in to fill that space. So you can see it's already starting to get more green because these other lipids or these other proteins are moving around very freely all over the cell membrane. And that's because those lipids in the membrane don't form polymers, as I said before. They form associations, but they're moving around at all times. It's sort of like, I don't know, you can imagine like taking your finger in it and you can sort of stir it around if you wanted to, if you were very, very small. But you can see from this, I don't show a lot of data in this course, but basically you bleach it and then there's no fluorescence right here. And then slowly those other proteins and lipids start to move into that region. And then you get the original intensity in that region. So lateral diffusion, so proteins and lipids moving side to side, happens very rapidly. However, flip-flopping, so a lipid head right here, and then switching to the other side, to now it'd be right here, that does not occur very quickly. And that's because of the interactions between those positively charged heads and those hydrophobic tails and their interactions with the water and stuff like that. So proteins and lipids do not flip-flop in membranes very quickly, but they do move from side to side and through the membrane very, very quickly for the most part. And so the cell membrane provides a barrier from the external environment to the internal environment. However, the cell does need to bring in molecules from the external environment to the internal environment. And so those proteins within the cell membrane, there's a bunch of different ones that are going to bring in and kick out a bunch of different molecules that the cell needs to survive. So things like ions, just molecules that you need, those building blocks, glucose to break down for energy. Your cell needs to be able to bring these in from the environment. And some of these just happen through diffusion, so they just go through protein channels. On some of them, you have to use energy basically to get them in the cell. But basically, here is a depiction of osmosis. So you have a high concentration of this molecule outside the cell and a low concentration inside the cell. So eventually, just through what's called Brownian motion, so just the motion of molecules, this will sort of even out if there's a way for that molecule to get into the cell. Now, this sort of limits how big a cell can get. So we are, I'm about 5'11" and I have, trillions and trillions of cells in my body. I'm just pulling that number out of thin air, but there's a lot of cells in my body. Why aren't I just made out of one big cell? Why isn't there a cell that we know of that's basically the size of a basketball or a watermelon? It's due to this fact. So you have basically a surface to volume ratio. And once that gets too big, so if you had a cell that was as big as a watermelon, it wouldn't be able to bring in enough molecules that it needs through that surface area to basically fill up that internal space and to provide for all of that volume within the cell. So that limits how big cells can get. So the largest cell known is actually, this is a really cool sort of image, but it's actually the axon or a neuron from the giant squid, which is like 12 meters long. So that's the biggest cell that's known, but it's actually very, very thin. Like it's very, very skinny, a few microns in diameter at some point. So that has no problem sort of bringing in solutes, but that's the largest cell that's known in nature. And then the smallest cell are these mycoplasma bacteria. And so those are ubiquitous. They're on your body. They can cause disease, but a lot of times they don't cause any problems at all. But these are actually the smallest cells that are known. So membrane transport and just the diffusion, the movement of molecules across a barrier like the lipid membrane, that limits how big cells can get. So how do we know so much about cells, and how do we now as people in, the 2020s know things that like the cell is the fundamental unit of life? Well, humans did not know that cells existed or that microscopic organisms existed for many, many years. So ancient people in ancient times, I think you can trace it back. There's like Greek writings where people hypothesized that there might have been creatures that you could not see that were causing disease, but they really didn't have any ways to visualize that or to prove those hypotheses. So that was not known until the microscope was invented in the around the 1500s, I believe. So the microscope is what allows humans to see microscopic cells. So they're too small to be seen with the naked eye. So we'll talk a little bit about microscopes, but microscopes are basically just telescopes in reverse. So telescopes were developed first, notably by Galileo and other people were making lenses in Europe at the time. And microscopes are basically just, you have to do some modifications, but Galileo actually apparently reversed his telescope and, changed it so that he could see microscopic organisms and stuff like that. He didn't really record a lot of it or spend a lot of time. He was more interested in the stars, apparently. And the lenses weren't sort of sophisticated, and the apparatuses weren't sophisticated enough to see things in great detail like we do right here. But so here's an image from the James Webb Telescope, and here is a really cool image of a lot of different microscopic organisms in some pond scum. So our ability to understand life and cells in general relies upon this technology called the microscope. And so the first human who we know looked through a microscope and then wrote down and published his findings was this Dutch guy who was a businessman, a clothes maker, and an amateur scientist named Antony van Leeuwenhoek. I don't know if I'm pronouncing that right, but that's as close as I can get. And so that's this guy right here. And so other people were sort of playing around and developing some microscope technology, but he was the first one to really make what is kind of a modern, sophisticated light microscope, which is the replica of it is seen right here, which was capable of magnifying a specimen 300 times. And so this occurred in the late 1600s. And so he looked at all sorts of things. So he looked at sperm under the microscope and red blood cells and pond scum and anything he can sort of get his hands on. And he made detailed sort of drawings. So this is a drawing that he made of cork. And he actually submitted his findings to the Royal Society in England. And that's sort of how the birth of modern microscopic and modern cell biology started. So his ideas were improved upon by others, including a guy named Robert Hooke, who was the first one to look at cork under a microscope and see these, what he saw, this structure right here of these individual units right here. And so he was the first one to coin the term cells because he believed that these looked like the cells that monks lived in monasteries. And you can sort of see what he's talking about right here. So these are plant cells that this guy Robert Hooke was looking at. So these individuals were the first people to sort of start the cell biology and microbiology in the late 1600s was when it first started taking off. And so now, in the subsequent years, we've developed all sorts of really good microscope technology. So there's a lot of different types of microscopes. You can spend a lot of money on a microscope and get really, really high resolution and really, really beautiful images. So light microscopes are the most common type of microscopes. So these are just basically you're putting light, you're looking at a specimen and the light is moving through, and there's high magnification with very sophisticated sort of lens structures. So multiple lenses that can magnify and focus the light in these images. So you can see these tiny cells here. So anybody who's done any sort of cell culture, this is very familiar to them. There's also, it's been modified, and we have things like fluorescent microscopy, which is light microscopy, but also able to detect and visualize fluorescence of different at different colors, basically. And so how this works is basically you take different molecules that have fluorescence to them and will bind to different structures. So for instance, this molecule DAPI right here binds to the DNA in the nucleus of cells and stains them blue. So this is a really good way to visualize cells because you can see all the nuclei in the cells, which we'll talk about in a bit. And you can also take antibodies and bind them to other proteins, and then take a fluorescent antibody and bind it to that antibody, or you can do that in one step, basically. But in general, it allows you to visualize other molecules within the cell. So these are, actin molecules right here, some sort of filament molecules within the cell. And then this, you could also stain another structure. I'm not exactly sure what structure that is in these particular cells. Another one which gives very high resolution is electron microscopy. So there's a few different kinds of electron microscopy, but basically you're using tiny, tiny electrons to visualize very, very small specimens in very, very great detail. So you can see this cell here with these sort of protrusions coming off of it that are allowing that cell to adhere to this surface and possibly move around. So all three of these images are actually of the same cell type called HeLa cells, which are developed from a human cancer cell line, which we'll talk about a little later. But there's lots and lots of different types of microscopes and lots of different type of microscopy that are used for all different applications and are very important for actually visualizing the cell and watching what's going on in the cell and their behavior, their morphology, what molecules are interacting, and then also just fine structural features like you can see these, I think these are like Filopodia on this cell right here. So if you look at all the cells on Earth, they can basically be broken up into three different what are called domains. Now, this is debated and some people sort of make cuts at different spots, but for the most part, this is the most accepted model. So these three domains. But it's broken up into prokaryotes, which are bacteria and archaea, and eukaryotes. So the right here, so the archaea and the bacteria, these are both considered prokaryotes. And so for bacteria, you have a huge diversity in the types of different bacteria that exist. So you have things like spirochetes and gram-positive and chloroflexi and proteobacteria and cyanobacteria, all very different from each other, but all prokaryotes and all single-celled bacteria. You also have the archaea, which are also prokaryotes. They're also always single-celled, but they actually are not related. So based on, and we'll talk about this in a bit, but based on sort of biochemical and genetic analysis of, really detailed sort of looking into the archaea and like the sequence of their ribosomes and stuff like that, we know that these species are in a domain of their own. So they're as far away from bacteria as basically we are. But they're sort of known because they were first found as sort of living in these very extreme environments, like hot springs and sort of high salt environments and stuff like that. But they're actually ubiquitous in the environment, and they may be actually the most numerous cells on the planet. But anyway, so there's bacteria and archaea. Those are the prokaryotes. And then we have the eukaryotes, and that includes animals, fungi, and plants. So anything that you see in your day-to-day life, we are all made out of eukaryotic cells. So those are all multicellular organisms. Slime molds are kind of multicellular, kind of unicellular. And then these sort of groups right here are all single-cell eukaryotes. So you can have eukaryotic cells that are single-cell eukaryotes or multicellular eukaryotes like the animals, the fungi, and the plant. But in general, the most accepted model for this evolutionarily is that all cells on the planet can be broken up into these three domains: bacteria, archaea, and eukaryotes. So bacteria are prokaryotic cells, and bacteria generally exist as free-living, single-celled organisms, and they are absolutely ubiquitous in the environment. Bacteria are everywhere. So you hear of them when they cause disease and stuff, but there's gajillions of bacteria all over your body, all over every surface, all over every environment, the soil, the seawater. On everything that you look at is covered with bacteria, these very successful living single-celled organisms that are prokaryotic. So here is an outline of a bacteria cell right here, and they have a few features that make them quite interesting. So just like other cells, they have a cell membrane that surrounds the cell, and then they have what's called a nucleoid, which contains the DNA material. So they don't have a nucleus like in eukaryotic cells, which we'll talk about in a bit, but their DNA is sort of it can be sort of centralized in a specific region within the cell. So their cell membrane is actually quite sophisticated and unique. So they have a plasma membrane, but they also can contain a cell wall and a capsule and other things that are quite unique to bacteria. So there's some features of their cell wall that's sort of unique to bacteria. Obviously, they have a cytoplasm, so that would be this region right here. And then the ribosomes, which basically, so this DNA is going to get transcribed into RNA, and then the ribosomes are going to be responsible for translating that RNA into protein. They're in the same cytoplasm as the DNA. So we talked a little bit about that in the other lecture. But the ribosomes are basically right by the DNA. So the DNA you can make RNA from the DNA, and then that RNA, even while it's being transcribed, can be translated into proteins in bacteria. So this allows them to do this very quickly. They also, some bacteria have a flagellum. So that would be this thing right here, which allows them to move around. And some bacteria can have things like piluses, which are these also used for motility, and it can extend outside of the bacteria and allow them to move around. Now, a really important thing about prokaryotic bacteria is that they are much, much smaller than eukaryotic cells that you would have in your body. So this image shows a few different bacteria that are invading a cell culture cell. And so this cell is going to be much larger than any of these individual bacteria. So they're estimated very much smaller, like 100 times smaller than eukaryotic cells. So that's prokaryotic cells, which are bacteria, which are prokaryotic cells. Now, I mentioned that bacteria are everywhere, so they're ubiquitous in the environment. And one place that they're especially important is in the human, what's called the microbiome. So the microbiome is the population of all microorganisms, including bacteria and archaea, but also fungal cells and stuff like that, but especially bacteria that exist in the human body, basically all over the human body. So they're especially prevalent in the intestinal places like the intestinal tract, all over your skin, in the oral mucosa, in your conjunctiva of your eye. They really are all over. And recent work has estimated that there's probably about as many, or if not more, microorganisms, including these bacteria, on your body as there are your own cells. So you are basically, human plus bacteria and other microorganisms. So most of the human microbiome is benign or beneficial. So most of these bacteria are not going to hurt you at all, and some can actually help do things like aid in digestion and also destroy other bacteria that may wish to cause you harm. So there's sometimes the microbiome can get sort of out of whack. So there's a certain bacteria called C. diff. I forget the first ones, but it's basically that bacteria. That's sort of the shortened name for it. But it can cause terrible sort of disease manifestations in your gut. And there's recent work showing that things like fecal transplants, so actually putting someone else's fecal materia with all their beneficial bacteria into a patient that's suffering from a C. diff infection, can actually be very beneficial. And so you've probably all heard, and there's a lot of benefits to eating things like yogurt or sauerkraut and stuff like that has a lot of these beneficial bacteria to get them into your gut. And so they can help you digest food and also help, keep other bacteria or fungi or stuff like that can cause problems in your gut and other parts of your body from taking a hold. So the human microbiome, it's a very important part of our biology and a very active field of research. So a lot of people are spending a lot of time looking at the microbiome on different areas of your body or different organisms' body and seeing how that contributes and how disease might arise from the balance in that microbiome getting out of whack. So bacteria can also be really useful for a lot of other things. And one example is this bacteria, Thermus aquaticus, and its relation to PCR, or the polymerase chain reaction, which we talked about in the previous lecture. So without this bacteria, Thermus aquaticus, PCR, which is the ability to replicate a specific part of DNA, a specific region of DNA in the lab, just using some reagents and a thermocycler. This would not be possible without this bacteria, Thermus aquaticus. So Thermus aquaticus was discovered in hot springs, I believe in Yellowstone National Park. So it's an extremophile. So it can live in very high temperatures and very extreme conditions. Now, what's really nice about that is that for PCR to work, remember we talked about how you have a piece of DNA and then you need to separate it. So in your body, you have different enzymes that are able to separate the DNA using energy from ATP. But in the lab, you just heat it up, and then you have two strands that are separated, and then you add the primer, and then you can build other strands off of that. Now, the problem with this is that the polymerases in your body that are actually going to synthesize this DNA on this strand are not going to be able to function at the high temperatures that you need to separate those strands of DNA. And if you drop the temperature down to where they can function, that DNA is just going to anneal back together. It's going to come together, and then you're not going to be able to do your PCR. So what scientists discovered is that Thermus aquaticus has this, since it lives at such high temperatures, it has a DNA polymerase, which is called Taq polymerase, that can actually function at very high temperatures. So this Taq polymerase was the first polymerase used in PCR, and it was integral to its success. So since then, there's been other modifications to this Taq polymerase to make it a little bit higher fidelity, so there's less errors in your PCR. But this Thermus aquaticus bacteria is essential for modern molecular biology and biochemistry. Now, I mentioned that bacteria can be very beneficial in your microbiome, or at least benign, and then there also can be used for a lot of different, beneficial applications and stuff. But of course, there are a lot of bacteria that cause disease in humans and cause great, suffering and death. And one of these is cholera. So cholera is a disease caused by this bacteria right here called Vibrio cholera. And so these Vibrio cholera cells basically get into your intestines, and they excrete a toxin called just cholera toxin that basically commandeers your signaling pathways and tells your intestinal cells to just put out a bunch of water, basically just to release water into your intestines. And what this causes is a potentially fatal diarrheal disease. So when you have cholera, so you're basically just excreting all of this liquid from your gut. And so the diarrhea that comes out is actually really weird diarrhea, and it sort of looks like rice water is what it's described as. But if left untreated, this will straight up kill you. And it caused many, many outbreaks over many, many years throughout human history. And it still plagues some societies today. So it was back in ancient times, they described diseases similar to cholera, and it can still cause up to 130,000 deaths per year today, mostly in the developed world. And the reason for that is that cholera is spread by basically poor sanitation. So people drinking water that has fecal material from people who have cholera. And so in regions where they don't have good wastewater treatment, and separation of wastewater from drinking water, you still can get cholera infections. So that's cholera. It is a bacteria that is definitely not beneficial and that you do not want to get because it can cause the horrible, fatal diarrheal disease. And interestingly, the bacteria cholera actually kicked off what is called modern epidemiology. And so epidemiology is basically people who track the spread of disease in an attempt to quell the spread of those diseases. And the person who's considered the father of modern epidemiology is this guy here, John Snow. So he lived in the 1800s, and he was an English physician, and he's considered basically the founder of modern epidemiology because of his work in cholera. So there was a really bad cholera outbreak when he was practicing medicine in Soho, London. And what he did was he tracked all the places where people were getting cholera And even as importantly, he tracked the regions where people were not getting cholera. So why is cholera in this region right here? So he said, okay, well, these people are getting cholera. I believe that it's probably coming from water that they're drinking. So people even back then did not know that cholera was necessarily spread through contaminated water. So he said, okay, no, everybody in this region is getting cholera, and everybody in this region is drinking from the same pump that's here on this broad street. And so what he did was very ceremoniously took the handle off of that specific pump, and guess what? The cholera completely went away. And so that is basically the type of work that epidemiologists do today to try and quell the spread of disease. And there's actually a sort of ceremonial pump at that region in London today in honor of the work that he did. Now, the other group of prokaryotic cells are called archaea. And so archaea are prokaryotic cells that were once considered bacteria, but on further examination, especially on really detailed molecular, genetic, and biochemical analysis, it was determined that they are their own domain. So they're something like as far from bacteria as we are from bacteria. They're their own domain. They may actually be a bit more closely related to eukaryotes. So this was determined via a lot of different ways. So like their ribosomes are very, very distinct based on the sequences. I mean, not that distinct, but they're distinct enough to be their own domain. And they also have some features like these lipids in their membranes have this weird sort of structure here. They're sort of esters that have these interesting sort of hydrocarbon things. So archaea, and they can basically look just like bacteria, but some of them have these sort of weird morphologies, like this archaea here that has this kind of cubic or square morphology. So they were first discovered in really extreme environments, like in these hot springs in Yellowstone, but also in environments that are very, have a lot of salt in them or have a very high or a very low pH. But since then, further research has shown that archaea basically exist in every environment on the planet. And so some evidence looking at seawater actually shows that they may be among the most abundant organisms on the planet. And so they're just that's just a feature of some of them, that they live in these extreme environments. So we're not going to go into a lot of more detail on archaea, but they're really important prokaryotic cells on the planet. So now that brings us to the eukaryotic cells. So animal cells, so all the cells that make up your body and all other animals are eukaryotic cells. And so eukaryotic cells are much larger and they're more complicated than prokaryotes. So one thing is that, and I just said, is that they're a lot larger. So this is an animal cell here, and here's just a really rough scale of a bacteria cell. It might even be a lot smaller than that. That would sort of be the largest that a bacteria would be in comparison to a eukaryotic cell. So very, very small prokaryotes are in comparison to eukaryotic cells. And another thing that makes eukaryotic cells very unique is that they have a sophisticated network of membrane-bound organelles. And so these are distinct membrane-bound regions within the cell that do specific functions within the cell. So one of these so we have sort of an electron microscope of a eukaryotic cell here, and we have a sort of cartoon diagram here. So eukaryotic cells all have, most of them, not red blood cells or sperm, but most of them have a nucleus. And so the nucleus is going to contain the genetic material. So that's going to contain the DNA. And so that nucleus has a double membrane around it, and that membrane is actually continuous with another organelle called the endoplasmic reticulum. So that's shown right here. So the endoplasmic reticulum is the site of lipid synthesis and also protein synthesis and protein folding and stuff like that. The eukaryotic cells also have something called a Golgi complex, and so that's involved in transporting lipids and proteins from the endoplasmic reticulum outside to other parts in the cells. And so to do that, eukaryotic cells use these secretory vesicles that can transport those materials out. Eukaryotic cells can also bring materials in through secretory vesicles and then degrade them in these things called lysosomes. And one really interesting feature of eukaryotic cells that is related to prokaryotes but really distinct from prokaryotes is they have these organelles called mitochondria. Mitochondria are really important. They're the powerhouses of the cell because most of the metabolism, most of the ATP that's synthesized in eukaryotic cells come from these mitochondria. And there's a lot of evidence to suggest that mitochondria actually arose from an endosymbiotic event where basically these prokaryotic cells got taken up and lived symbiotically within these eukaryotic cells. And so there's a lot of evidence in support of that. Mitochondria actually carry some of their own genetic material, and so they have their own genes within mitochondria. So we'll talk a bit more about that in a bit. But these eukaryotic cells, they're much larger and more sophisticated than prokaryotic cells, so the bacteria and the archaea. So plant cells and fungi, I'm not going to talk about fungi specifically, but plant cells are also eukaryotic cells. So their cells are more like animal cells. So they have a lot of things in common with animal cells. So they also have a nucleus, endoplasmic reticulum, Golgi complex. They have these secretory vesicles and lysosomes and stuff like that. They also have mitochondria. But plant cells are distinct in a few different ways. So one is that they have a cell wall that's really rich in that polysaccharide we talked about in lecture 4 called cellulose. So this gives plant cells a lot more structural integrity than animal cells in general. And so that's one thing that's interesting about them. Plant cells usually contain a very large vacuole. And so this can contain water and other biomolecules that is sort of important for maintaining the turbidity or the structure of the cell. And it can also have, different molecules that are important for the cell's function or the feature of the cell in it as well. The other thing that plant cells have that's very important is they have these membrane-bound organelles called chloroplasts. And so chloroplasts are the site of photosynthesis. So they're similar to mitochondria, although they are different. So mitochondria are believed to come from a prokaryotic bacteria. Chloroplasts are believed to come from like a prokaryotic cyanobacteria or photosynthetic prokaryote that was taken up into plant cells and then sort of lived within them. So photosynthesis, the conversion of sunlight into energy, and then that energy used to build carbohydrates in plants, that takes place in the chloroplast. So the chloroplast is the site of photosynthesis. So it's sort of the most important powerhouse of the plant cell, whereas the mitochondria is the most important powerhouse of animal cells. So a lot of people mistakenly think that eukaryotes are always multicellular organisms like plants, animals, and fungi. But actually, there's a lot of eukaryotes that are single-cell organisms. Some of them may live in these sort of colonies that sort of work together, but a lot of them just exist as free-floating, organisms in their own right, but they're still eukaryotic cells. So these single-celled organisms are sort of placed in a really nebulous classification called Protista or Protozoa or protists. So that's sort of a catch-all term for single-celled eukaryotes. So they're not bacteria, they're not archaea, and they're not multicellular, so they're protists or a protozoa. These species are highly diverse, so they may be as distantly related to each other as we are from them, and they have a lot of beneficial and there's a lot of disease-causing species within the single-celled eukaryotes as well. So this is an amoeba, which is a single-celled eukaryote that sort of has this interesting structure. Yeast are single-celled fungi, actually, but they're single-celled eukaryotes because fungi are eukaryotes, and so they're beneficial in some ways and not beneficial in other ways. So this is an image of a trypanosome, which I'll talk about in a bit. But this is a single-cell eukaryotic parasite. And so there are several single-celled eukaryotic parasites that can cause disease, including malaria. But this is one of them that can cause African sleeping sickness or Chagas disease. There's also single-celled eukaryotes that perform photosynthesis. So algae are single-celled eukaryotes, like this Volvox algae right here. This is Giardia, which I hope you never get. So that's a single-celled eukaryote that causes terrible intestinal issues. So if you're ever out west, don't drink from the water in the lakes or the rivers, because there's a good chance that it has Giardia, and then no one's going to want to share a tent with you. And then this is a Toxoplasma gondii, which sort of forms these really cool structures right here. But it actually causes toxoplasmosis, which is carried by, or toxoplasma, which is carried by cats. So a lot of times they tell pregnant women not to go around cats because you can get, toxoplasmosis. It's basically not a problem unless you're immune compromised. So a lot of HIV/AIDS patients actually die from toxoplasmosis. So there's a good chance you yourself have been exposed to this single-celled eukaryote, toxoplasmosis, but your immune system is able to keep it in check. But people with compromised immune systems can have problems with it. So a lot of single-celled eukaryotes, very diverse family, very interesting species. So not all eukaryotes are multicellular organisms. So I just mentioned one of these single-celled eukaryotes are called trypanosomes. And so some of these unicellular eukaryotes do cause disease. So one of them is this trypanosome here, which I forget the scientific names of them, but this one is carried by tsetse flies in Africa, and it can cause African sleeping sickness. This is a really terrible disease to get. Another one is this trypanosome here, which is carried by this bug. It's actually a true bug. It's sometimes called the kissing bug. And so it causes something called Chagas disease in South America. And so these are really diseases that you do not want to get. The treatments for them are not great. There's no vaccines for them. So there's a lot of different, basically the best way to avoid them would be to sort of set up these traps, which are used to collect tsetse flies so they do not bite people. And then in terms of these kissing bugs, a lot of times in South America, they're sort of drawn to houses that aren't so well insulated. So they're attracted, and they can live very easily in things like thatched roofs or sort of adobe walls with cracks in them. And then at nighttime, they're called kissing bugs because they actually come in and feed on you. And then the bite isn't what spreads the trypanosome. They actually, when they feed on you, they will also defecate. And then you scratch the sort of itch or the bite that they gave you, and you sort of rub some of that fecal material into the cut. And then that's how you would get the trypanosome and the Chagas disease. So these single-celled eukaryotes are very, very important for infectious disease. So now we can start talking about multicellular eukaryotes. So I have a bit of a mistake here. Not all animals, plants, and fungi are multicellular. All animals, most plants, and most fungi, like yeast, aren't multicellular. But let's just say most animals, plants, and fungi are multicellular. That is, they have many different types of cells with specialized functions that work together to comprise a single living organism. So we have the blue whale here. That is the largest organism that's ever known to have lived. We have a bunch of plants here, and then we have some mushrooms here, which are funguses. So these are all multicellular eukaryotes. Now, interestingly, multicellularity in eukaryotes is estimated by people who work on this to have occurred at least 25 times in natural history, which is pretty interesting. And there's a whole bunch of different hypotheses as to how that actually occurred. So one of them is something like this colonial flagellate hypothesis. And a lot of them are sort of based on this sort of idea. So basically you had a unicellular protist that is a unicellular eukaryotic organism. And then that unicellular protist or eukaryote would sometimes make sort of an aggregation at certain times under certain conditions. And this does happen with a lot of different unicellular eukaryotes all the time. So this would have something happen like with these flagella being sort of them interacting to form more of a coordinated sort of organism. And then you would get something like specialized eukaryotic single cells that would do a specific job. And then that could result in something like invaginations that are sort of the basis of how multicellular eukaryotes develop from, a zygote or a single cell into a complex multicellular eukaryote with these sort of invaginations and these specialized cells. So there's a lot of different theories onto how this occurs. I am not an expert in that at all. But needless to say, when we think of most organisms like animals, plants, and fungi, we think about multicellular individuals, and all multicellular species are eukaryotic, have eukaryotic cells. And you can sort of see from that last example these hypotheses on how multicellular organisms arose evolutionarily is sort of mimicked when you look at the actual development of multicellular organisms from a single zygote, which would be, a sperm and an egg coming together, through into an adult organism. So you have, and again, I'm not an expert in developmental biology. It's a really interesting subfield of biology with a lot of different work on it. It's its whole own field. But basically, you have this single organism, and then that splits into two. You have these very specific events, so cleavage, and then that goes into two cells, then to four cells, and then you can form what is a blastocyst, basically. And so that's a more specialized group of these cells. And then you get these sort of invaginations called a gastrula. And that's the beginning of the sort of formation of these multicellular organisms that all have these three germ layers, which we'll talk about in a bit. So all multicellular organisms have an ectoderm, a mesoderm, and an endoderm, and then they sort of have a cavity in the middle, which would be your sort of digestive tract. And so all the different cells in your body are part of either the ectoderm, the mesoderm, or the endoderm as that arose during development. And then that will progress into a juvenile stage and then finally into an adult with all of these specialized cells that came specifically from this differentiation early in development. So like I said, this is a really rich, very interesting field. And there's most of the work on it uses a few different model organisms. So this Xenopus frog is a really useful organism because their eggs are really large and easy to study. Zebrafish are also one that's used quite frequently. Same with fruit flies, Drosophila, and worm C. elegans, where basically every cell in the body has been mapped out. There's something like 900 cells, or there's a very specific number of cells in C. elegans worms, and basically these developmental researchers can trace each one of those cells through its development. So it's a very rich field, very interesting topic, very big topic, so we can only scratch the surface here. So animals, not humans specifically, but animals have the most complex diversity in cell types compared to plants and fungi. So multicellular animals have about 100 to 150 different cell types, whereas plants and fungi have about 10 to 20. So that's quite interesting. And each of these cell types, as I mentioned before, comes from those stages in your development, from the ectoderm, the mesoderm, or the endoderm that occurs after gastrulation. And so the ectoderm is sort of the external layer, so that would form the epidermis, your skin cells, neurons, and also things like pigment cells, also a lot of other different cell types. This is just sort of a survey of them. The mesoderm includes your muscle cells, so cardiac muscles, skeletal cells, and then smooth muscle in the gut. Also red blood cells and tubule cells in the kidney. And then the endoderm, which is the internal layer, makes alveolar lung cells, thyroid cells, and things like pancreatic cells. So each one of your cells in your body can be traced back to this event early in your development. And so I briefly mentioned it before, but now we can actually go through the organelles that make up eukaryotic cells, because these are quite important and very interesting. So they have these, eukaryotic cells have these very distinct membrane-bound organelles. So these are compartments within the cell that each do specific functions for the cell as a whole. So this is not found in prokaryotic cells. It's basically the main difference between eukaryotic cells and prokaryotic cells. But here we have a diagram of an animal cell and all the different organelles within it. So of course, we're going to go through pretty much each of these very quickly. So we have a nucleus and then a nucleolus, which is a subset of the nucleus, which you've actually probably already seen without knowing it in some of the microscopy pictures I've shown. So the membrane of the nucleus is actually continuous with the endoplasmic reticulum, which is the site of a lot of protein production, protein folding, protein sorting, and also lipid production. So there's a rough endoplasmic reticulum, which is where proteins are sort of formed, and then a smooth endoplasmic reticulum where lipids are formed more readily. There's also a Golgi apparatus and also all sorts of different vesicles that are involved in getting proteins sorted and brought to the position where they need to be, whether that's secreted out of the cell or delivered to a membrane within the cell or delivered to a specific compartment within the cell. There also may be vacuoles in animal cells, although those are more common in plant cells. There are lysosomes, which break down material that's brought into the cell, and peroxisomes, which we're not going to talk about, but are sort of related to that. There are also various cytoskeletal filaments. So we have microfilaments, microtubules, and intermediate filaments. And so these are the cytoskeleton. They're sort of the structural skeleton of the cell. So they give it its shape and sort of structural integrity. And they also do things like transport stuff around the cell. So they actually work as sort of highways to transport both vesicles like the peroxisome or lysosome or endosomes, and specific, proteins, and they are responsible for movement in the cell as well. There's a picture of a centrosome here, which is involved in cell replication. We also see the plasma membrane here, and then we see probably the most interesting of the organelles, arguably, or at least I think so, or the most unique, which is the mitochondria, which are the powerhouse of the cells where energy is produced in the form of ATP. And then, of course, surrounding the entire cell, we have the plasma membrane. And then all of the contents inside the cell is considered the cytosol. That's actually the aqueous environment between the plasma membrane and the membrane separating these other organelles. So that would be called the cytoplasm or the cytosol. And that's where soluble proteins and all of these organelles are going to exist. So these are the cellular organelles. We'll go through each of them very quickly and talk a little bit about the different jobs that they do and their structure and functions. So we'll start off with the nucleus and then this structure within the nucleus called the nucleolus. And so this is often called the brain of the cell, but it's not making conscious decisions, and it's not actually like part of the nervous system or anything like that specifically. It's called the brain of the cell because it contains most of the DNA in eukaryotic cells. So most of the DNA is stored within this nucleus here. So it's actually surrounded by a double membrane, a nuclear envelope. So this is sort of a specific membrane called a nuclear envelope, and that partitions the inside contents of the nucleus from the rest of the cell. However, certain molecules, especially stuff like RNA, needs to be transported out of the nucleus to be, to undergo translation. And so that's where these nuclear pores come in. So here's sort of a spherical picture of the nucleus. So right here you see it as a flat, but it would actually be something like a sphere. And you can see these little pores that are in the nuclear membrane, and these are very important. And so here's sort of a close-up. And so that's really important to get materials in and out of the nucleus. So if you look at microscope images like this electron microscope image here, this is the nucleus all around here. And then there's usually often this very unique region that's stained very darkly, and that's called the nucleolus. And so that's the site of rRNA or ribosome assembly. Ribosomes are made up of partially RNA and partially protein, which I've mentioned a bit before, which is quite interesting. And this rRNA and these ribosomes are actually synthesized and assembled in this specific region right here called the nucleolus. And so if you're ever doing something like light microscopy in cells, you'll always see this, you'll often see this sort of blue circle here. And so that is a stain called DAPI that will specifically stain the nucleus in cells. And so this makes it really easy to sort of visualize these cells because you can always orient yourself with the nucleus. So that's the nucleus. It's the quote unquote brain of the cell because it contains most of the DNA in eukaryotic cells. And by most, I mean the vast majority. The mitochondria contains some DNA, but almost all of your genetic information is stored in the nucleus. Now, we saw in the previous image that the nucleus actually has usually, even when you stain it in cells, this very distinct circular or oblong or oval structure or shape. So that arises because of the nuclear lamina. And so this is a structure, it's a structural protein that maintains the assembly of the integrity of the nuclear membrane and the pores. So in this image right here, so this would be the nuclear envelope. And then you have this structural protein, which is the nuclear lamina, that's sort of holding it all together. And so, unfortunately, some individuals have diseases called laminopathies, where the lamina is not, there's a mutation or something in the lamina that causes issues. So this can lead to diseases including progeria, which there's a lot of different types of progeria, but they're defined by basically rapid aging in individuals. So individuals are born, they have sort of distinct features and distinct medical issues, and then they usually do not, they basically can resemble, depending on what type of progeria that you have, they can resemble old people, like very elderly people by like the age of 20, and they do not live very long. So it's a very devastating disease. And it's a laminopathy, so it's a mutation or an issue with the lamin that maintains the nuclear envelope. And so here is stained green is a normal nucleus, so this is the lamin from a healthy individual, and then here is the lamin from an individual with a specific type of progeria, and you can see how it's sort of just falling apart. And this causes all sorts of problems and leads to early fatalities, unfortunately. And so the next organelle we're going to talk about is the endoplasmic reticulum, sometimes just called the ER. And so the endoplasmic reticulum is actually continuous. So it's a membrane-bound organelle, but it's actually continuous with the nuclear membrane, which you can kind of see here. And so these nuclear pores will deliver RNA to the endoplasmic reticulum. And there's actually two types of endoplasmic reticulum. There's a rough endoplasmic reticulum and a smooth endoplasmic reticulum. So here is an electron micrograph of a rough endoplasmic reticulum, and it's called the rough endoplasmic reticulum because it's got all these little dark spots all over, and it just sort of has like a rough appearance. So those dark spots are actually ribosomes. So ribosomes are part RNA, part protein, all protein-making machine. So they are basically the factories where you take mRNA, this different type of RNA called tRNA, and then we talked about this before, and that's how you get translation. So you get that polypeptide chain growing from those ribosomes. So the rough endoplasmic reticulum is the site of a lot of different protein synthesis, protein processing. So sometimes different parts of proteins need to be cut off from the other parts in order for them to be functional, and also folding. So a lot of protein folding has to occur in the rough endoplasmic reticulum. There's also a smooth endoplasmic reticulum that's often it's often located in a different part of the cell, but it's somewhat continuous with the rough endoplasmic reticulum. But you can see it has a much smoother appearance. And so this part of the endoplasmic reticulum is involved in lipid synthesis, and both the endoplasmic reticulums are involved in cholesterol synthesis. So the lipids from the endoplasmic reticulum will ultimately often be transported to the plasma membrane or other organelles in the cell. So that's the endoplasmic reticulum. It's really important because it's the site of a lot of different protein synthesis, processing, and folding. Now, there's also this other really interesting organelle in the cell that's got a really distinct look about it, and it's called the Golgi apparatus. So it's named after the guy who discovered it, Dr. Golgi. And so you see it, it's always depicted as having this sort of weird shape that's full of these blobs. And you can actually see a really good electron micrograph of it here. And it's basically these folded like a sandwich, these different layers of lipid bilayer membranes within the cell. So what does this Golgi apparatus do? Well, it's involved in transporting these proteins from the cell and from the endoplasmic reticulum and then getting them to where they need to go in the body. So it's part of what's called the secretory system in the cell. So you have proteins that are being synthesized in the rough endoplasmic reticulum. They're going to be transported through vesicles to this side of the Golgi apparatus, which I believe is called the cis side. And then those proteins are processed and sorted through the Golgi apparatus. And then they move to the trans side of the Golgi apparatus. And then they'll be put into these secretory vesicles. And so they can be delivered to different parts of the cell, or they can actually they can fuse with the cellular membrane, and those proteins can be secreted outside of the cell. So a lot of different cell types secrete proteins in multicellular organisms, like signaling proteins or neuropeptides, stuff like that are secreted outside the cell. So the Golgi complex is involved in determining which proteins go where in the cell. So that's also a really interesting field and how the cell is actually able to achieve that task. So that's the Golgi apparatus, and it's part of the secretory system in determining where proteins go when in the cell. So sort of opposite from the Golgi apparatus, cells also have these mechanisms where they obviously need to bring in material from the external environment and then process those within the cell. So these are through processes of endocytosis. There's also phagocytosis, and these both involve structures, organelles within the cell called lysosomes. So proteins, materials, and even entire organisms like prokaryotic bacteria can be imported into the cell, and then those can be broken down and the cell can use those individual building blocks. So we have a really nice diagram down here. There's a protein or a group of proteins, and it can interact with the cell membrane. And then that extracellular material, there's actually an invagination in the plasma membrane. And then that will bud off and actually form its own vesicle. So I remember we looked at SARS-CoV-2 virus and influenza virus entry into the cell. So this is the type of process that would operate through. There's also processes, some cells, especially like immune cells called macrophages, are really good at this process called phagocytosis, where they can actually use this same process to bring in entire organisms like bacteria and then engulf them and then break them down in the cell. Now, how the cell breaks down both these like protein components or just parts of the extracellular environment or these entire particles, it basically treats them like food. And so this is outlined in this figure right here. So we have a food particle right here, and the cell is going to start engulfing that, and then it'll form basically ultimately a vesicle, and it will fuse with these other membrane-bound organelles called lysosomes. And so these are very low pH, and they contain a bunch of digestive enzymes, and they'll fuse with that food that was taken in, and they'll end up breaking down those molecules. So this is a really important process for cells to basically get nutrients and building blocks and biomolecules from the external environment, break them down, and then use them for their own processes. Now, unfortunately, there's about over 70 characterized diseases that are called lysosomal storage diseases. And these are caused by mutations in those enzymes within the lysosome that are supposed to break down these molecules so the cell can use them for other purposes. So what happens if you have one of these diseases, an enzyme's not going to be able to break those molecules down, and then your cell is going to be basically stuck with them. It doesn't know what to do with them, and that causes all sorts of problems in your cell. And depending on which enzyme is mutated, depends on how the disease is going to manifest. So 2 examples of this is one is Tay-Sachs disease, and so that is a result of a mutation in an enzyme that breaks down this molecule right here, which is a ganglioside. And so this enzyme will actually break these bonds here, or specific bonds in there, in the lysosome so that you can use that molecule for something else. Unfortunately, if you have a mutation, that leads to buildup and then really, really bad disease manifestations. Another one is Hunter syndrome, which is the result of a mutation in this enzyme right here. And now your body's not able to break down these molecules called glycosaminoglycans, or GAGs, which are these really long, the most famous ones are like heparin sulfate, which you might have heard of. And so there's these really long carbohydrate-based molecules that have these negatively charged sulfur groups that are involved in the extracellular matrix in your cell. So if your body can't break these down, again, it causes really bad diseases. Now, these are inherited diseases. They're both autosomal recessive, so they're a recessive gene, so you have to inherit one from a carrier father and one from a carrier mother. So they're actually really rare in the general population. However, in certain communities that basically are more related to each other because they're sort of closed off from other societies, like Ashkenazi Jewish populations or Amish populations, or certain Quebec French populations or Cajun populations, these diseases are way more prevalent in children born into those populations because you're more likely to be somewhat closely related, like a cousin or a second cousin to someone, and that mutation got into that population at some point in history. And then because they're closed off communities, these mutations are much more likely. So these are lysosomal storage diseases, and they happen when cells are not able to break down certain biomolecules that they receive through endocytosis or phagocytosis because they're not able to break them in this organelle called the lysosome. Now, one of the coolest organelles, I think, are mitochondria. So mitochondria are called the powerhouses of the cell, and that's because they're the site of most of the metabolic activity in eukaryotic cells. So this is where carbohydrates are basically finally turned into a whole bunch of ATP, which is usable energy for other parts of the cell. So there's mitochondria. Here's an electron microscope image of a mitochondria here. And so they actually are double membraned organelles. And you can see that they have an outer membrane and they have an inner membrane that sort of folds into itself to make these features called cristae. And you can see it a little better in this cartoon here. So they have this outer membrane, and then they have this inner membrane that forms these invaginations called cristae. And so what that does is it increases the surface area of a bunch of different enzymes and proteins that are involved in metabolic processes in the mitochondria. So it increases the amount of energy that you can get out of one mitochondria. So here is another image of how this sort of happens. So here is basically the outer mitochondrial membrane it would be, and here's the inner mitochondrial membrane. And so your body takes in fatty acids, glucose, and amino acids, and through a process that occurs in the cytosol of the cell, so outside of the mitochondria, it turns these into acetyl-CoA. Well, actually, it doesn't matter. There's a little caveat to that, but basically acetyl-CoA is the starting point, and that gets into the mitochondria, and then that enters the citric acid cycle, which produces, and then the electrons are taken off that acetyl-CoA, and that provides energy to power something called the electron transport chain. And what that does is it uses that energy to pump protons, so H plus ions, which we talked about, into the inner membrane space. So this space between the inner membrane and the outer membrane of the mitochondria. And so that makes a gradient. It makes more protons in that intermembrane space than inside of the mitochondria. And so there's proteins that can use that proton gradient as a pump to power these molecular machines called ATP synthase that synthesize ATP in the mitochondria. And that ATP is the universal energy currency of all living things. And so the mitochondria are really good, really important for making a bunch of ATP for your cells. So they make most of the ATP that you use to do anything in your cells that I'm using to talk to you right now. And so mitochondria are also really cool because they actually resemble prokaryotic cells, and there's a lot of evidence that suggests that they are an ancient endosymbiotic prokaryote that basically lives within eukaryotes. And now eukaryotic cells basically can't live without mitochondria, and then mitochondria can't live without eukaryotic cells. So these are really cool organelles that are really important for your cell's function. And so some of the cells in your body actually have more mitochondria than other cells, including something called brown adipose cells that are in brown adipose tissue. So that would be right here. So this is a brown adipose cell. And this is a white adipose cell. So in your adipose tissue, this is basically fat tissues or different fat tissues. So these white adipose cells, most of your cells, especially as adults, are white adipose cells. So each of these cells contains one really big lipid droplet that has all these triacylglycerol fats that you can use for energy, especially under stuff like starvation conditions. Now, brown adipose cells are a little different because they can have more than one smaller lipid droplet, and they have a bunch of mitochondria, which is what gives them this darker color. You can see some of these mitochondria here. So brown adipose tissue is more prevalent in newborns and in hibernating animals, but it decreases, the amount of brown adipose tissue in your cells decreases in mammals as they age. And so the brown adipose tissue is actually really good for thermoregulation. So all those mitochondria breaking down all that fat molecule provide a lot of heat. So it actually can keep these hibernating animals from basically going into hypothermia and also newborn infants. So it's a pretty interesting type of cell that has a lot more mitochondria than other types of cells. And so that fat broken down in brown adipose tissue, as I said, provides heat, and it also provides water to these hibernating animals during the winter. So here's a groundhog right here, and here's a black bear right here. They both hibernate or semi-hibernate, and during that, they'll break down those brown adipose tissues so that those fat stores can keep them alive for quite a long time. There's also, I don't know if any of you have seen this show, I Shouldn't Be Alive, I think is what it's called, but it's basically a show where they talk about people who survived really treacherous sort of experience where they basically should have died, but they got lucky and sort of got out of it. Well, there was one many years back that told this true story about this couple who got stranded in the snow with their newborn baby. And it was a really scary episode to watch because what they basically ended up doing was putting the baby in a suit jacket container case or whatever, and then dragging it through the snow. And they were very, very worried. And there was this horrible moment where they opened it up and they were so scared that, the baby wasn't going to make it. And the baby was perfectly fine. And so they sort of cut to a scientist, and what kept the baby alive was that it had a lot of brown adipose tissue, and that was actually provided an extra amount of heat for that newborn infant that actually kept it from going into hypothermia. So it's a pretty cool feature that mammals have evolved to have this sort of brown adipose tissue to help basically thermoregulation and protect against hypothermia. Now, chloroplasts are kind of the equivalent to mitochondria in some ways in plant cells. So whereas mitochondria are the powerhouses of animal cells and all eukaryotic cells, really, chloroplasts are the powerhouses of plants and algae cells. So they're very similar to mitochondria. So here's a plant cell here, and you can see they'll contain several of these chloroplasts. So they're similar to mitochondria in that they also have an inner and an outer membrane in these chloroplasts. And they contain these thylakoid membranes and these different structures that basically, to make a long story short, these are where the light reactive molecules and reactions are going to take place. So this contains chlorophyll and these different proteins, these photosystems, and these are going to absorb these light molecules and use the energy of that light to, just like in mitochondria, pump protons to make a proton gradient on one side of the membrane as opposed to the other side of the membrane. And then that proton gradient is then used by the same molecular machine, this enzyme called ATP synthase, to synthesize ADP from ATP. So here's a electron micrograph of these sort of structures within the chloroplast. But these chloroplasts are the powerhouses of plants and algae cells, and they're quite similar to mitochondria, including the fact that there's also evidence that they arose from an endosymbiotic event to where a photosynthetic prokaryote basically was absorbed by a eukaryotic cell, and now the chloroplast can't survive without the plant cells, and the plant cells can't survive without the chloroplasts. So those are chloroplasts, another very interesting organelle. And then plants also have these pretty cool structures called vacuoles. And so these are quite important for several reasons, mostly because they contain a lot of water and other small molecules, and they provide sort of a turgid pressure against this cell wall that sort of has all this cellulose and can sort of push into the cell. So they give the cell a lot of integrity. They can also contain specific molecules that give specific cells specific features. So for instance, there's these anthocyanin molecules shown here in purple that are within the vacuoles of these plant cells here. And this can give color to different specific cells in specific plants. And for instance, like when you eat citrus fruit, the vacuoles contain that citric acid in them. So they keep that citric acid away from the rest of the cell, so it's not going to affect the rest of the cell, but that's what you're tasting when you basically eat an orange or a lemon or a lime. So the cytoskeleton isn't really an organelle, but it's a really important part of the cell. So the cytoskeleton, these are different protein components that are in eukaryotic cells that they basically extend from the nucleus to the plasma membrane and give structural support to this cell. So that's a really important thing. So they maintain this cell shape. They also are involved in trafficking of different vesicles and proteins throughout the cell. So they provide structure, but they also are like providing roads basically throughout the cell. But we'll talk about that in a minute. So there's three main types in eukaryotic cells: microfilaments, intermediate filaments, and microtubules. And they're basically different protein structures, and they have different functions in terms of their structural ability. But for the most part, they tend to form these really strong, long fibrils that are made up of smaller units. And so here is, I believe, an intermediate filament, and then this is a microtubule. So they can sort of grow and recede very quickly because they're made up of these aggregations of these smaller units. So that's the cytoskeleton, and it gives structural support and shape to the cell. And somewhat related to the cytoskeleton is the ability for cells to move. So a lot of different cells require basically motility. So they have to actually physically move around the environment. And through evolutionary history, different cells have evolved many different ways to actually move throughout their environment. So the flagella is a really common way that cells can move through. And so this is actually just like a molecular motor. So if you look at this picture right here, there's all these different proteins that are interacting the outside of the cell and inside of the cell to actually make something that can actually move, and it's powered by ATP. And so here is an algae cell. You can see it has 2 flagella. Some animals have 1 flagella. Some animals have more flagella. And then prokaryotic flagella actually move in a different way than eukaryotic flagella. But cells can also move just through the action of their cytoskeleton. So the cytoskeleton can actually, get bigger, get smaller, sort of interact with things in the environment. It can make these sort of things coming off called lamellipodia, and then these filopodia. And so this can allow cells to sort of interact with and move through their environment. So these are very common in things like macrophages that move around the cell and use these to engulf other cells like bacteria for phagocytosis. And the cytoskeleton is also really involved in cellular trafficking. So I'm not going to go into huge detail on this, but all these vesicles and all these transportation of all these things through the cell that we've been talking about, those have to be moved somehow. And how it happens is basically there's these really interesting motor proteins that will attach one side to these vesicles and another side to these types of filaments or cytoskeletal elements within your cell. And they use those as basically like railways to transport vesicles and other processes through the body. This is also sort of how your muscles move in a more complicated fashion. But cellular trafficking is really important. So I had an undergraduate teacher who said, basically, the secret to life is getting the right protein to the right place at the right time in the right concentration. He really thought like a biochemist or a cell biologist. And through this process of cellular trafficking, that's how this is achieved. So we already talked about it a little bit, but cell division is a really important feature of cells as well. So specifically, we're just talking about eukaryotic cells. So they will grow and divide a lot of times based on extracellular signals that will initiate a bunch of different complicated reactions within the cell. It's a really sort of complicated process leading to mitosis or meiosis. And this involves intracellular enzymes, chromosomes, the membranes, and all sorts of things that are basically the proteins and DNA within the cell all coordinate to have cell division. So there's this sort of cell cycle that is shown right here. So there's sort of a senescent phase, and then once a cell gets a signal to divide, it'll go into a G1 phase, and then it'll go through to a S phase or synthesis phase, where it synthesizes new DNA. Then it goes through the G2 phase, and then mitosis actually occurs. So we'll talk a bit more about this in the next lecture because we're going to be talking about reproduction. And so there's an interphase where the cell is basically just normal, that nothing's really happening. And then there's a prophase where basically it starts to form this mitotic spindle, which are those cytoskeletal microtubules, and they connect to these things called centrosomes, which are shown right here. And then there's changes in the chromosomes, the DNA in the nucleus, so they sort of condense into these chromosomes that can actually be replicated. Then there's a prometaphase where they sort of line up and you get this spindle going on here with these microtubules. And then those chromosomes will line up in the middle of the cell and be connected to that spindle right there on either side. And then it goes into anaphase, where those spindles actually pull this separate the same chromosomes, so there's two copies in each cell. It'll actually pull those apart. And then the last phase is telophase and cytokinesis, where actually the two different cells will separate. And so here's a really cool image here where you can see a bunch of different cells and they're in different phases. So this would be like in prophase here. This one is in what would be like anaphase, and same with this one. So if you look under a microscope, it's pretty cool. You can actually see all different types of cells if you have growth factors, which are those extracellular factors that initiate replication. When you look through the cells and you sort of stain them, you could see these sort of structures, which is pretty neat. So that's cell division. So it's really fundamental to eukaryotic cells and prokaryotic cells. But eukaryotic cells use this process here. Now, it's really important that cells can divide. However, the cell needs to be able to control that growth very carefully because uncontrolled cell growth can lead to cell proliferation, and then that can develop into what are called neoplasms or tumors that can also metastasize and develop into cancer. So again, this is a really big topic, and there's a lot of different cellular processes that are involved in cancer biology and tumor development. But I'll just mention one of them here. So there's a lot of growth factors that basically tell cells, okay, it's time to grow and divide. So you have like insulin will do that, epidermal growth factor, VEGF, that's a really common one. And so these growth factors will bind to receptors on the cell, and then there'll be a complicated series of interactions between proteins and changes in the cell that'll ultimately lead to cell growth and possibly differentiation. But cell growth is what we're talking about. Now, there's some proteins like this protein whose structure is shown here, which is a pretty cool protein called PTEN. And so PTEN is actually a tumor suppressor gene because it shuts off these pathways. And that's really important because that would stop the cell. So, okay, the cell needs to grow, but we don't want it to grow and grow and grow and grow and grow because that's how you develop tumors. So something like PTEN tells those pathways when to stop growing. Now, however, so it makes a lot of sense that something like PTEN is mutated in a lot of different cancers because if there's mutations in PTEN and it's not functioning properly, it's not going to tell the cell to stop growing. So the cell is just going to keep growing and growing and growing. And that's how you can get tumors like this tumor right here from a colon tissue. So again, it's a very big topic, but the fields of cell division and cancer biology are really intermingled. Now, there is one really, really useful application for science that comes from cells that sort of grow uncontrollably, and that's cell culture. So the ability for scientists to culture different cells in the lab that grow on a plate and just keep growing is really, really useful. So these cell culture uses a lot of times what's called immortalized cell lines. So normal cells, if you just take them and put them on a plate and grow them, they'll grow a couple times, but then they'll die. Cells aren't supposed to grow all that much. So you can develop cell lines that have certain mutations. They're basically tumor cells. But they can be maintained in a dish. So basically what you do is you would take those tumor cells and you would put them in a petri dish and you would add some media. So that media is going to contain things like nutrients. It's going to contain buffers to keep the pH at a certain level, and it's also going to contain growth factors. So the normal one is like fetal bovine serum. So you get serum from cow fetuses that have a lot of growth factors in them. So that's going to tell these cells to keep growing, and they're just going to keep growing and stop growing and not stop growing. And so you can get cells like this so that you can see under the microscope. And so they basically get to what would be called confluency. So they're basically all the cells are going to be touching each other. So these cells are about, I'd say about 80 to 90% confluent. That's something you just get used to working with cell culture. And then you basically remove them from the bottom of the dish and then split them into like 2 or 4 dishes. And then they keep growing and growing and growing. And so this is a really fundamental part of biochemistry and cell biology. And it's a really fundamental part of biochemistry and cell biology because there's a ton of different applications and uses for cell culture. So cell culture has really revolutionized. So it was first started in the 1950s. There was no immortalized cell culture lines before that. And it really revolutionized molecular biology, biochemistry, and also medicine and agricultural research. So nowadays, there's a huge variety of different cell lines, immortalized cell lines. So if you're studying lung cells or like lung disease, you can get lung cancer immortalized cell lines. If you're studying liver cells, you can get liver cells, all sorts of different cell lines from all sorts of different species. And they can be used for a lot of different things. So you can get a bunch of them together and use that for protein production. A lot of vaccines are produced. So these are attenuated viruses. You would just passage them on cell culture and they'd lose some of their virulence. And so they can be used for vaccines. A lot of vaccines are produced in cell culture. If you work in a virus lab, cell culture is really important because you'd study and grow the viruses on cell culture. So this is a plaque assay here. So these spots that you can see here are where the cells, the virus actually infected the cells and then killed those cells. And you can visualize how many viruses are in your sample by how many of these plaques or these little spots are there. That fluorescent microscopy I showed you, you can study different cellular processes by using microscopy in cell culture. And so that's really important. And there's even starting to be like cultured meat. So that's sort of a dream. Hopefully we won't have to kill cows someday to make steak. We can just use cell culture. That's very, sophisticated. We're not there yet. But this particular hamburger that I'm showing you right here was actually made by cell culture. And it's the same proteins that are in, a hamburger that you would get from a cow, but you don't have to kill a cow. So cell culture is incredibly important in cell biology, molecular biology, biochemistry, and also medicine and agriculture. And so I mentioned that cell culture was not established until 1951. Many people tried. They would take all sorts of cells and put them on plates and try and get them to grow, try everything they could, and these cells would just die after a couple times. So the first immortalized cell line developed for cell culture was this cell line called HeLa cells. And so they were established in 1951 at Johns Hopkins. And so these HeLa cells were actually taken from a cervical tumor from a 30-year-old woman who died that same year, and her name was Henrietta Lacks. And so for years and so that's where the name comes from. HeLa is for Henrietta, and HE is for Henrietta, and LA is for Lacks. For many, many years, it was basically lost to, most scientists had no idea where the name HeLa came from. There was a lot of publications that was just like, I don't know, that seems to be lost in history. Well, they came from this specific woman. And so I don't know about most, but many, many scientists, myself included, have worked with HeLa cells that came from this woman who was an African-American woman who living in the United States. She had 5 kids. And who died at a very young age from this cervical tumor. But those cells that came from her body really revolutionized medicine. However, this book was released pretty recently, within the last 10, 15 years, called The Immortal Life of Henrietta Lacks, where this woman, Rebecca Skloot, actually investigated where these cells came from, and she sort of uncovered this entire story of Henrietta Lacks. And she went and talked to her family, and they had absolutely no idea that their grandmother's cells were being used in, all of biology and biochemistry. So it's kind of a really interesting story, and it brings up a ton of different bioethical issues, basically, all revolving around informed consent. So she did not necessarily give her consent for her cells to be used. And then her and her family, although this has benefited biomedicine and science and tons of companies, those companies making, vaccines and stuff on these cell lines are making money hand over fist. But there really wasn't any, compensation to her or her family, or even any acknowledgement from it. So it's a very sad story, but it's a very complicated bioethical issue. So if you have time, I definitely recommend reading this book and looking into that story. And it's just sort of finally becoming, I think they made a movie about it on HBO, but it's just sort of finally coming to light, this whole story, based on the efforts of this woman who investigated it, to the point where there's now like statues of Henrietta Lacks honoring her and her, unfortunate contribution to revolutionizing science, really. So Henrietta Lacks and HeLa cells, so it's all related to cell biology and cancer and sort of society and the bioethical issues surrounding it. So now some other processes that are really important to cells are involve the proteasome and autophagy. And so these are processes where Basically, the proteins in your cells have a set lifespan. So there are some proteins in the lenses of your eye that were basically there when you were born, but all the other proteins in your body and a lot of the cells as well have a set lifespan. So they don't exist forever. If they did, the cell would be all clouded up. There could be problems with them and stuff like that. So your body has your cells have developed these mechanisms to sort of recycle these proteins and break them down and then use those amino acids to make more proteins. And so this is a very active part of research and very important for a lot of different processes. So one of these processes is the ubiquitination system. So basically that's outlined right here. It's too much detail. We don't need to go into it. But basically, there's these proteins that will tag these proteins. So this right here is the protein, and then it puts these little tags called ubiquitin, and they're called ubiquitin because when they were first investigating it, they were very ubiquitous in the cell. There's just a lot of these little proteins, and so once this protein gets tagged by this small protein molecule called ubiquitin, so it's shown right here. That protein that's attached to ubiquitin will then go into this really big, complicated protein machine called a proteasome, and that'll actually degrade and break down the protein into its amino acid components, so those can be reused to make other proteins. There's also a process called autophagy, which is related to, like phagocytosis and endocytosis, which we talked about earlier. But basically, that's when the cell is basically eating itself. So it'll basically take big swaths of material, including entire mitochondria, and enclose them in this organelle called an autophagosome. And so all sorts of different molecules, like I said, even mitochondria, will be put into these vesicles, and then they'll fuse with these lysosomes that we discussed earlier. And then the lysosomal enzymes will break down all the components in that autophagosome. So that's a way for your cell to sort of clear old debris from it. And it's a really active form of research. Everything we've been talking about has a ton of people working to uncover the secrets of it. But the proteasome and autophagy are quite interesting processes that the cell uses to recycle old proteins and other components, including organelles. And now for the next slide, I'm just going to warn you, I have a trigger warning here, and this is so if you have arachnophobia like me, the next slide is going to have a picture of a big scary spider, but they're pretty actually gentle spiders. So if you're worried about that, then please cover the left side of the slide coming up in the next slide. So I know I'd appreciate that. So so the next slide, I'm going to show a spider. So what happens when a cell dies, basically? So we talked about cells, growing and differentiating and cell division and everything. What happens at the end of a cell's life? So one thing that you don't want to happen to a cell is necrosis. So necrosis is what happens when there's an injury to your cells that basically causes the cell to lyse. And it's going to spill all those contents within the cell, including proteases and all sorts of molecules, into the surrounding area. And that can affect other cells nearby and cause injuries. So there's a lot of different ways that necrosis can occur. One of them is very applicable to people who live in Kansas. I have an old house. It's got an attic. It's got a basement. And every once in a while, I see this guy right here, which is a brown recluse spider. They're actually pretty gentle spiders. They do not want to mess with you. That's why they're called recluse spiders. I don't like killing spiders, even though I have a natural fear of spiders. I am arachnophobic, which apparently is something that sort of is just in some people from birth. They've done some studies. But I try not to kill spiders because they're good. They eat other bugs and stuff like that. And it's sort of, if you're from the Midwest, it's sort of bad luck. But I do kill brown recluse spiders because their bites can be quite bad, and I have pets and I don't want them to get hurt. So this brown recluse spider bite can cause significant necrosis of tissue if you do get bit. Like I said, it's not easy to get bit by them. It usually happens if you like don't put your clothes in the, like you just have your clothes on the floor, and then these brown recluse spiders like to hide during the day. So maybe they'll like sneak into a pant leg, and then you put the pants on it and then it goes against your skin and bites you. I've actually been about to go outside and I was just going to throw my shoes on because I'm just turning out for a second. And then I put one shoe on, and then I went to put the other shoe on, and something in my gut told me, hey, I better check this shoe. And then when I flipped it over, and a brown recluse spider came falling out of it. So if you're in Kansas or anywhere else where brown recluses are around, just keep an eye out for them. But because they can cause terrible injury, so that is where this injury on the right came from. But this happens from necrosis, which is when your cell, like I said, when your cells lyse and spill their contents into the surrounding area and can cause these sort of injuries. Now, the better way for a cell to end its life is through a process called apoptosis, and that's programmed cell death. And so this is a really interesting feature of multicellular organisms, and it's involved in development and just the normal processes in your body. So in certain organs, they all have a certain period of cell turnover. So like your gut, those cells are only going to be alive for like a week or two or something like that. And then they're going to undergo apoptosis. Now, why apoptosis is preferable to necrosis is because it's a cell death where the contents of the cells do not just spill out into the surrounding area. So it's a step-by-step process where basically you have a cell that's programmed or it has some extracellular message that says you need to die, basically. And so what'll happen is the nucleus is going to fragment, and then all these changes are going to happen, and you're going to form these apoptotic membrane protrusions right here. And so basically you get these little blebs right here, these little membranes that contain the contents of the cell. And these organelles within these can actually be fully functional and everything. So these little cells, or these little blebs, or apoptotic bodies as they're called, can actually be phagocytosed by other cells, and then those components of those cells can be used by other cells. The building blocks can be used. So apoptosis is a preferable way. It's the way your cells are supposed to die when they've reached the end of their lifespan, rather than necrosis. All right, so that's the end of lecture number 5, which was all about different types of cells. So I hope that you found it interesting and learned what a cell is and why it's so important. It really is the fundamental unit of life. So I hope you understand what it's made out of. So what a prokaryote is, like a bacteria or an archaea, versus a eukaryote. So there's single-celled eukaryotes, and then plants, fungi, and animals are all eukaryotic cells. Hope you found some of the different processes that cells do pretty interesting, like cell replication, apoptosis, protein trafficking, the cytoskeleton, stuff like that. And then we talked about some diseases associated with that as well. And then we talked about the different organelles within eukaryotic cells. So the nucleus, the endoplasmic reticulum, the Golgi, and the mitochondria and chloroplasts and stuff like that. So I hope you found it interesting. Please let me know, of course, if you have any questions. And so remember also to finish your quiz for the end of the week and do your short essay for this week, which I believe will be on slime molds, which are very interesting sort of species. So thank you very much. I hope you enjoyed.